We have done, and others have done, a significant investigation of genome in myeloma. Most of the studies have focused on the expressed genes, exome sequencing, for example. Now, the expressed genes constitute 2% of the genome. The rest of the 98% of the genome has not been studied. So this investigation, which we just presented at ASH, focuses on the whole genome, 100%, with a special emphasis on the 98% which has not been studied, and probably, in our opinion, plays a very critical role in causing the disease and the driving the disease. So what we find is that overall, doing a very deep sequencing, what we call ATX, we find 11,000 mutations in each patient on an average. Now, we have analyzed 200 patient sample. This is our collaborative work with the French group, the IFM-DFCI study, and these patient samples have been studied with all possible important sequencing platforms. We have done whole genome sequence, we have done RNA sequencing, we have done microRNA sequencing, copy number alterations, even epigenome. And our presentation focused on this 98% of non-coding area, and we find that there are certain hotspots which are frequently involved. Promoter region, what we call super enhancer regions. And in this analysis, we have identified a lot of new translocation, which might be driving the disease in subgroup of patients. We have also found that certain type of processes might be causing this mutation, and they would become target for our treatment in future, and very importantly, could be target for prevention. And then we have found areas which are outside of the gene, but affects the function of the gene. So we have identified a gene called BCL7, a gene called BCL6. Their abnormality outside of the region is present in almost 70 to 80% of the patients. So it's a very common phenomenon. We think it happens early, and those are maybe playing a very important role in how maybe MGUS and smoldering myeloma develops and how myeloma behaves. So now our focus is to understand what these genes do and how can we counteract that effect with the mutational process. So to summarize our study, it is the first deep look at the whole genome, so the real myeloma, and not surprisingly have identified many interesting and important areas that may be playing a critical role in genesis of myeloma and also later progression of myeloma. I think our study is going to provide us new leads for treatment and hopefully prevention of the disease.